Good morning, students. In the last class, we were studying about the pollen-pistil interaction. Yes or no? We studied about the pollen-pistil interaction, that how does this takes place? From the time the pollen comes and sits on the stigmatic surface, yes, the stigma of the flower, you all know very well which is the part of the stigma, the stigmatic surface, the pollen comes and sits. It's all imaginative, students. See, here is the stigma surface and these are the pollen grains. So what happens here when the pollen grain comes and sits on the stigmatic surface? Yes, till the pollen grain germinate on the stigmatic surface, forms a pollen tube and enters to the megasporangium, that is the ovule of the flower. Until there it is known as pollen pistil interaction. It is known as pollen-pistil interaction. Till last class, we studied about this. The definition about pollen-pistil interaction is important. Pollen-pistil interaction definition is important. Now, what happens in this pollen-pistil interaction? There is a recognition of compatible pollen. Yes, whether this pollen grain is of this flower only, whether this flower whether this pollen grain is compatible to grow, means whether it can adjust there, recognize, and it can grow the pollen tube. I had given you the example last time that of the phone. Your phone has got a face recognition. Yes or no? Your phone has got that face recognition. When your phone recognizes your face, it unlocks. Yes or no? It unlocks. So what happens here? It recognizes. Yes, the similar way here also, the pollen grains grow onto the stigmatic surface, form a pollen tube. Yes, form a pollen tube, and the pollen, that is the male gamete, enters inside the megasporangium, that is the ovule of the flower. Ovule of the flower, fertilizing the egg. Yes, fertilizing the egg. So this is nothing but the pollen pistil interaction. Now we will study, students, in today's class, what are the advantages of this? What are the advantages of pollen pistil interaction? Means what are the benefits we get with this pollen pistil interaction? Okay, interaction is nothing but meeting. Yes, meeting where the pollen and the pistil meet. That interaction. What are the advantage? What is the benefit? It helps in the pollen recognition. Yes, it helps in the pollen recognition, whether this pollen is of this flower only, whether it is of sunflower, whether it is of hibiscus, whether it is of rose, whether it is of jasmine. It recognizes. Okay, it doesn't allow any and everything pollen grain to grow on it. It only allows the growth of those pollen which are compatible, Okay, so this is one of the advantage. Helps for plant breeders to get desired hybrids. Yes, what happens in this pollen pistil interaction? When we allow the pollen grains of our desired pollen grains, which the pollen grains which we want to grow on it. Okay, the pollen grains which we want to grow on it, those pollen grains only can be allowed to germinate, so helps for the plant breeders to get desired hybrids. For example, if I want to hybrid two of the plants, okay, we will be studying it in the further artificial hybridization. We are going to study it in the further artificial hybridization. There you will get a very clear picture. But before that, let me explain the second point. So what happens here? Helps for the plant breeders to get a desired hybrids. Means, for example, if they want to get a desired fruit or a desired vegetable of a hybrid quality, of the that quality which has got less seeds, or which has got large flesh in it. So something like that, where they want to get a new variety of fruits, new variety of vegetables. Okay, new variety of fruits, new variety of vegetables. So it helps these plant breeders, the one who cross the plants. Okay, they help this plant breeders to get a desired hybrids, means to get a new variety, of plants. Okay, next third point is helps to study the pollen germination on stigma. Yes, it helps to study the pollen germination on stigma. How the pollen will germinate onto the stigmatic surface. You all know the stigma, uh, on the stigmatic surface, the pollen grain grows on it. Yes, it germinates and forms one pollen tube in this way. So these are the stigmatic surface. See students here. So this is the stigmatic surface. Yes. So what happens on this stigmatic surface? The pollen grain will come, place itself into the stigmatic surface, and then germinate with the presence of the sugar syrup and the nutrient present on the stigma and form one pollen tube. Okay, what happens here? The stigmatic surface, okay, the stigma surface here, what happens? The pollen grain will germinate or grow, forming a pollen tube on the stigmatic surface because of the presence of the nutrients on the stigma. Presence of the nutrients on the stigma. So this is nothing but these are the advantages. The first advantage is nothing but pollen recognition. It recognizes the compatible pollen, whether this pollen can be grown or not, or whether it can germinate or not. The first one. Second one, plant breeders to get desired hybrids. Yes, they get desired hybrids, means their own quality, new variety of the plants or the vegetables or the fruits. Helps to study the pollen germination on stigma. Yes, I just now I had shown you, right, the stigmatic surface where the pollen grains grows and it forms a pollen tube. So this is about the advantages of pollen pistil interaction. Next, coming to artificial hybridization. Just now I had told you, right? What is this artificial hybridization? Okay, students, before going to all this explanation part, okay, before going to all this explanation part, let me just give you one overview. What happens here? So we take one plant. Okay, now for example, any one of you, for example, if I take a Purnima or Daneshwari or Sonal or Bharat, Deepak or anyone here are the scientists who are working in the this hybridization. Okay, or Karthik, anyone who are working on this hybridization, artificial hybridization. Now, what they will do here, they will take one plant. Okay, any plant they want to. For example, if they want to take paddy, if they want to take any other fruit or any other vegetable plant, so what they do, they take the plant. Later they do the process known as emasculation. Now what do you mean by this emasculation? Emasculation is nothing but the removal of the male part of the plant or the flower. The removal of the male part, and you know what it is? It is the removal of the anther. Removal of the anther from the plant is nothing but called as emasculation. Okay, removal of the anther part of the flower is nothing but called as emasculation. Now what they do, they remove the anther part of the plant. And why they do this? This whole process is artificial hybridization, students. First, they take their own desired plant. They remove the anther part. Okay, they remove the anther part. Once they remove the anther, later they bag the plant or the flower. They do this bagging process like this. Can you see here? They do this bagging process. The bagging can be done with a bag made up of butter paper. So what they do, they bag this flower once after removing the anther. That process is known as emasculation. That is the removal of anther. That process is known as emasculation. After emasculation, they bag the flowers. Bagging is done by the using a butter paper bag. Okay, a butter paper bag. So after bagging, after once bagging, later, after once the bagging is done and it is kept for some more time. And why the bagging is done, this question can be asked. Why the bagging is done? And you also must be very curious to know it. Bagging is done to prevent the contamination of stigma from unwanted pollen. Okay, to prevent the contamination of stigma from unwanted pollen. Now, why it is told that to prevent the contamination? Because no other pollen grain should come and pollinate it. No other pollen grain should come and pollinate it. So because of that, the bagging is done. The question can be asked, why emasculated flowers are bagged? Emasculated flowers are bagged or covered to prevent the contamination from other pollen grains, to prevent the contamination from the other unwanted pollen grains. Once the stigma surface matures, stigma surface matures, what happens? The desired pollen grains, the pollen grains which we want to, the pollen grains which you require, you people who are doing this, whichever you feel that, okay, this is the pollen grain, I want to pollinate this plant, that is being dusted on the stigmatic surface. You place it on the stigmatic surface. And after placing it on the stigmatic surface, you once again bag it and allow the fruits to develop. Allow the fruits to develop. After bagging, after bagging, once the stigma surface is mature, once the stigma surface is mature, you once again bag the flower, you once again bag the flower and allow the fruit to grow. You once again bag the flower and allow the fruit or the vegetable to grow. Now, why all this is done? Why this all process is done? So, it is done to cross between varieties, different varieties of plants, species. Okay, to get a new variety, to get a new variety and a desired variety. Desired means which you want, which plant you want, which new plant you want to make, or a new variety you want to get. Okay, desired pollen grains for pollination, stigma protected from contamination. So why artificial hybridization is done, and what is this artificial hybridization? Artificial hybridization is nothing but crossing different varieties of species, crossing different varieties of species. Okay, crossing different varieties of species, for example hibiscus varieties, hibiscus and brinjal can be, or a gawa and any other fruit. Then it can be between the jasmine or something like that, where we cross different varieties. Okay, different varieties. Once we do crossing is done, that is to get one new hybrid variety, to get a new hybrid variety and a desired variety. Desired means which you want to make, which you want to invent, or which you want to discover. So that can be, for that reason the hybridization is done. Okay, now first step here is emasculation. You remove the anther part. Once you choose the plants you want, you remove the anther part, and that part is known as emasculation, and it is very important process. Plus it is an important definition also. Emasculation is an important definition. And what is emasculation? Emasculation is nothing but removal of anther. That is the removal of the pollen grains. Removal of the pollen grains. Emasculation is nothing but removal of anther. Next is bagging. Bagging, why it is done? Bagging is nothing but covering by a butter paper bag, covering with a butter paper bag to prevent the contamination of the stigma by any other pollen grains. To prevent the contamination of stigma from any other pollen grains. Once the bagged flower, the stigma becomes mature, you allow the pollen grains to grow on it, the desired pollen grains, the pollen grains which you want to pollinate. Later, once again, it is bagged, and it is allowed to grow the fruits. It is allowed to grow the fruits. So this process is important, students. The artificial hybridization definition can be asked. Explain the artificial hybridization can be asked. Explain the processes of the artificial hybridization can be asked. Clear? You need to give this emasculation, bagging, and what is done in this entire artificial hybridization and why it is done also. So it is one of the important questions, can be asked for 3 marks, definition for 2 marks, and also for to explain entirely for 5 marks as well. So remember this. So remember this. What and all is done? You remember one picture. You remember one picture in your hand, one imaginary picture. So here what happens? First you choose the flowers. First you choose the plant. Remove the anther, that is emasculation. After removing the anther, it is bagged. Once it is bagged, and why it is bagged? So that it is not contaminated with any other pollen grains. Once it is once the stigma becomes receptive, that it becomes mature, you dust the pollen grains on it. Yes, you dust the pollen grains on the stigma. Once again, bag the flowers and allow the fruits to develop. Allow the fruits to develop. So this is nothing but artificial hybridization process, and it is a very important, it is a very important process, students. So remember, you need to study this, one of the important processes, that is our artificial hybridization. Next, coming to post-fertilization events. Next, coming to post-fertilization events. So until now, we were studying only about the pre-fertilization events. Yes, then the fertilization events. Now we have come to the post-fertilization events. So what is this post-fertilization events? Post-fertilization events is nothing but the events which takes place after fertilization. The events which occur after fertilization or after the double fertilization. Post-fertilization events, all the events that occur in a flower after double fertilization is known, or the fertilization process is known as post-fertilization event. So it is an important definition, can be asked. What is a post-fertilization event? Post-fertilization events are nothing but the events which occur in flower after the fertilization process. After the fertilization process. And what are the major events in them? Development of endosperm, first one. Development of endosperm, development of embryo, maturation of ovule into seed, maturation of ovary into fruit. So development of endosperm, the first one. And you know endosperm is a nutritive tissue. Development of embryo. Yes, after once the zygote develops into embryo, how the embryogenesis, that is the development of the embryo, takes place. Maturation of ovule into seed. Yes, how the ovule converts or forms or develops into seed, and how ovary develops into fruit. How ovary develops into fruit. So these all are the post fertilization events. So all these are the major post fertilization events. The first one, endosperm tissue. Now, let me make it very easy for you students. Now, what is this endosperm tissue? This endosperm tissue you must have seen in the coconut. The coconut white kernel part. Yes, the coconut water or from the tender coconut or this white kernel part is nothing but the endosperm tissue. The endosperm tissue. And how does this endosperm tissue is formed here? How does this nutritive tissue is formed? It is formed from the primary endosperm nucleus. It is formed from the primary endosperm nucleus. Pen that nourishes developing embryo, that provides nourishment to the developing embryo. And students, now how does this fertilization, means how does this formation of endosperm takes place? So this is the megasporangium. In that megasporangium there is an embryo sac, you remember. So there are antipodal cells, the synergids, the egg cell. Yes or no? And so in the double fertilization, what happens, students? In the double fertilization, the male gamete, one male gamete which fertilizes the egg, the other male gamete goes and fertilizes the central cell, that is the polar nuclei. Yes or no? The polar nuclei, forming a triploid primary endosperm nucleus. Forming a triploid primary endosperm nucleus, and that primary endosperm nucleus forms into endosperm tissue that nourishes, that means it provides the nourishment to the developing embryo, the embryo which has been growing, it provides nourishment to the developing embryo. And two types of endosperm development are there. How many types of endosperm development? Two types. One is free nuclear type, free nuclear endosperm development. Another one is cellular type endosperm development. There are two types. One is free nuclear type of endosperm development. Another one is cellular type of endosperm development. So students, first thing you need to remember in the endosperm, endosperm is a nourishment tissue. It is a nutritive tissue formed from the primary endosperm nucleus that nourishes the embryo, which is growing inside. Okay, that nourishes the embryo which is growing inside. So it is an endosperm nucleus. It is an endosperm which is a nutritive tissue. After that, in coconut, it is the white kernel part. Yes, it is the white kernel part, and the coconut water from tender coconut, and the other one is the white kernel part is nothing but the endosperm. It is nothing but the endosperm. And there are two types of endosperm development. One is free nuclear type of endosperm development and cellular type of endosperm development. Yes, there are two types of endosperm development. One is free nuclear type of endosperm development and cellular type of endosperm development. Yes, and it is a nutritive tissue formed from the primary endosperm that nourishes the developing embryo. And if you have to know what is this endosperm, so it's nothing but the coconut water which is present in the coconut, and the white kernel part which is present here. The white kernel part which is present here is nothing but endosperm. And how does this endosperm is formed? Endosperm is formed by the double fertilization process, students. So what happens in the double fertilization process? The male gamete will come, and one male gamete will fertilize the egg, the other male gamete will fertilize the polar nucleus. When it fertilizes the polar nucleus, it forms the triploid endosperm nucleus. That triploid endosperm nucleus, or that triploid primary endosperm nucleus forms into endosperm, which nourishes the developing embryo, and there are two types of endosperm development. One is free nuclear type of endosperm development and cellular type of endosperm development. So this is about the post-fertilization events. So today we studied about the advantages of pollen pistil interaction. First one is pollen recognition. One is the advantage. Then the second one is to get a desired hybrid, to get a new variety of plants. And the third one is to study the pollen germination on the stigma. Yes, the pollen germination on the stigma. Second one is artificial hybridization. So first you need to remember what it is, artificial hybridization. Artificial hybridization is nothing but which is done by artificially by the human, manually by the humans. Manually humans pollinate the flowers. Manually humans pollinate the flowers. Okay, so crossing of the different variety means pollinating the different variety of flowers. Pollinating the different variety of the flowers is nothing but hybridization process. Now, how it is done here? It is done in very easily. For example, if I take, we have taken hibiscus flower here. Yes, we have taken hibiscus flower here. So what happens when we take a hibiscus flower? We first emasculate it. That means we remove the anther, pollen grain of that flower, and we bag it. We bag it in this way so that it is not contaminated with any pollen grain. Any pollen grain should not come and sit on the stigma surface and form the pollen tube. Yes, and form the pollen tube. Next, what we do? We take another pollen grain. Yes, we take another pollen grain, which is our desired one, which is our desired one. For example, I will take the pollen grain of datura. Yes, I take the pollen grain of datura. This flower is hibiscus. Clear? Now what we will do? Once the stigma becomes mature, we will dust the pollen grain of datura on the stigma surface of hibiscus. On the stigma surface of the hibiscus, and once again rebag it. So that there is no contamination again, and we allow it to develop into flower or the fruit. So this is nothing but the process of artificial hybridization, students. It is one of the important questions, time and again been asked in the question papers. Definition are asked, the different steps have been asked, so you need to remember the imagery, remember this image. Clear? Remember this image. And also this process you can write down and take so that it becomes easy for you. Clear? Okay. Second is post-fertilization events. What are the those events which takes place after the fertilization process? Major events are development of endosperm, development of embryo, maturation of ovule into seed, and maturation of ovary into fruit. So these are the different major post-fertilization events. Clear? Next, coming to endosperm. How the endosperm is formed? What is this endosperm? Endosperm is a nourishing tissue. There are two types of endosperm development. One is free nuclear type and one is cellular type. Okay, one is free nuclear type, one is cellular type. There are two types of endosperm development. And the example of the endosperm is coconut tender, coconut water, and the white kernel part of the coconut. And how it is formed? It is formed by the double fertilization process. Where the male gamete will fertilize the polar nuclei in the central cell, forming the triploid primary endosperm nucleus. So we studied till the endosperm today. We will studying further about the development of the endosperm as well, then the different other major events in the post-fertilization process. So today I'll be giving you one short assignment. That is the home assignment, and you need to write about the artificial hybridization, the definition, and the different events or the process which we do in the artificial hybridization process. So this is a home assignment, you know, where you have to submit it to me. I want all of you to submit it to me today itself. Before I do your next class, I want all your home assignments with me. Is it clear to all of you? Yes. So please make sure that you send your home assignments. Even if you have got any doubt, any confusion, anything, any particular topic, if you have not understood, you can personally inform it to me so that I can clear your doubts. So thank you and have a nice day, students.